If it is, again, we'll see about I'll just go to sleep.
All right, we're going to go ahead and get started. All right, you guys can quiet down. We'll get started now. Okay, so welcome to our Undergraduate Research Day, our annual event where we celebrate the undergraduate students that are doing research in our department. We have a whole day planned, starting out with our speaker. The next event from 10:30 to 12:30 will be poster presentations. I hope everyone is there. We'll have lunch. Oh, I think we have lunch. We'll have, please, we'll have some other visitors coming. Please take the time and, visit with our guest, and hopefully he'll be inspired by all the, great stuff that you're doing. And then in the afternoon, we have a panel session on how students can get involved in research. So probably for most of you, the panel session may not be so, you're already, probably everyone in here is already doing undergraduate research, and probably almost everyone in here is presenting a poster. But the panel session, we'll have some of you actually are going to be on the panel session, and so we'll talk about how the process of getting involved, undergraduate students getting involved in research. And then the poster sessions will be judged, so make sure that if you're presenting a poster, you'll each be judged by, I think, probably 3 judges, and so make sure that if you haven't, by the end, 12:30, if you haven't been judged by at least 3 judges, make sure you let us know. But I think it should go pretty smoothly. So I'm happy to introduce our speaker today, Dr. Tom Royston. He's a professor and head of biomedical engineering at the University of Illinois in Chicago since 2009. He's been a faculty member there since '95. He received a career award, NIH, NIBIB, NAGI Award, the Acoustical Society of America Lindsay Award. He's a fellow of the American Society of Mechanical Engineering and the American Institute for Medical and Biological Engineering and the American Society of America. He's a founding member of the National Society for Magnetic Resonance in Medicine, Magnetic Resonance Elastography Guidelines Committee. That's tough. And he's offered lots of papers and given a lot of presentations. I'm sure this is the most important one that he's ever given. So please help me welcome our speaker.
All right, thank you. And wow, standing room only. This is amazing on a Friday at 9 in the morning.
Getting food and,
Yeah, well, there you go. There you go. We do the same thing. We feed our students. Yeah. Okay, so no, I'm really happy to be here. I'm going to give a technical presentation. Also have a few slides starting about my department, university, and then afterwards I'm happy to, I've got some more slides, or I'm happy to answer any questions about my research or about UIC, biomedical engineering more generally, or Chicago. So yeah, thank you for the intro. And let me start first of all, from the Richard and Lawan Hill Department of Biomedical Engineering. I've been there a while. A few things about our department. I think it has, in some ways, it's very similar to FIU, though we have been around a bit longer. We're one of the first biomedical engineering programs in the country. It's been around since 1965, and it started as a collaboration between a couple of medical schools and a new engineering school in 1965. And over the years, it migrated into engineering, but then about 15 years ago, it went back to being both in engineering and medicine. So I report to the deans of engineering and the dean of medicine as head of biomedical engineering. And that's been really good for us in terms of fostering and supporting translational research and nice opportunities for our students. So we are a department that is about 30 core faculty, a number of affiliate faculty, mostly in medicine. We have about a pretty good sized PhD program, about 100 PhD students. We have a separate PhD program in bioinformatics, as well as in BME. About 80 MS students. I think we're about the same size undergraduate program as you, with about 340 Bachelor of Science students. And I'm perhaps looking out at the room here, I'm guessing we have some similarities in terms of diversity. Like many BME programs, we're about 50-50 men and women. And UIC is a minority serving institution. There is no majority ethnicity or race at UIC. We are designated as a Hispanic serving institution. Overall, the number of alumni we have at the BS, MS, and PhD levels. Okay, so that's, again, I'm going to talk more about the department later, but you're here bright and early on a Friday morning, so I'm going to try to teach you some technical things, tell you about what I do in research. And the first thing I probably should do is explain what is elastography, and then I'll talk about some of the applications we've focused on in muscle and cornea, which seem completely unrelated, but there are actually, they share some common challenges. So what is elastography? So elastography is built on the fact that you do have changes in mechanical properties, the stiffness, the viscosity or damping of tissue. You will have changes that will correlate with disease or injury, and also response to therapy. So we'd like to be able to measure and quantify these. One way to do this is with a biopsy, but that is invasive, and it's prone to sampling error. You're only pulling out a small piece of tissue. If you look back several thousand years, the technique of manual palpation has been used since, at least since the Greeks in ancient civilizations. Identify tumors, check for fibrosis. So elastography is based on these concepts, but it employs an imaging modality in some way to be able to see inside the body or see within layers on the surface of the body in a way that we can assess the stiffness of the tissue. So I've done research using ultrasound-based elastography, magnetic resonance-based elastography, and optical coherence or optical methods for elastography. These all have their pros and cons. Optical obviously is somewhat limited to the surface. Ultrasound has the advantage of being dirt cheap relative to MRI and being rather portable bedside. MRI, yeah, it's expensive, but it has the most overall capability in terms of pretty much seeing anywhere in the body and being able to, ultimately what we're doing is we're vibrating things, and we need to image how those vibrations are traveling through the tissue, and we're using that to identify properties. So one application that we're focused on is muscular degenerative diseases, which will, like muscular dystrophy, which will alter the mechanical properties of the muscle. Another application, sorry, some of these images might feel slightly disturbing in the morning, is the cornea, where there are different conditions or actually bad surgeries that will cause a weakening of the cornea. In the cornea, you have intraocular pressure in the globe of your eyeball, which can cause it to bulge. And then you have other conditions which cause a stiffening of the cornea. And the cornea needs to be somewhat compliant and flexible because it's very important in terms of how your eye focuses. Okay, so elastography, if we look at any type of solid, there are two basic types of mechanical waves that travel through it. One of these is we call longitudinal compression waves, and the other one would be transverse waves. Now the compression waves in the body, everywhere except for bone and lung, are pretty much all traveling at the same speed, around 1,500 meters per second, because your body is mostly water, and basically they're just telling you your body's mostly water. And these are ultrasound actually is based on sending in compression waves at really high frequencies up in the megahertz and getting reflections back. The transverse waves, those are different. They will be much more sensitive to the tissue connectivity and essentially the tissue elasticity. So we're more interested in studying these waves, and they're at a much lower frequency range that we use, down on the order of tens to hundreds to maybe up to 1,000 hertz or so. And so our focus is on those types of waves. And so elastography, whether it's MR based, ultrasound based, or optical based, there's basically three parts to it. Or you can think of it that way. You need to send in vibrations. There's different ways you can do this. We will use, sometimes you'll use like speakers, like vibrating speakers, shakers, depending on the application. There's all kinds of ways you can do this. You need some way to image the wave propagation. And I'm not going to get into the technical details of how we do this, but in MRI, you can, I have a couple of, well, there's some cool YouTube videos that kind of try to explain this. But basically, you encode a position on a particle in MRI, and then if you vibrate it and it moves, you can track that motion. It's incredibly sensitive. You can get down to being able to track microns of displacement using MRI. It's called MR elastography. So you get this image of wave propagation, and then you got to do some type of inverse model where you'll have the waves traveling, and you'll try to back out, well, what was the stiffness that created that wave pattern? So this is called the inverse problem or the reconstruction problem. Okay, so really simple. If you just think you have these waves traveling through, if the tissue is stiffer, the waves are going to be longer. If it's softer, they're going to be shorter, and it kind of goes by this simple equation, which is the square root of the shear elasticity divided by the density of the tissue. So we assume the density is pretty much always the same, around 1,000 kilograms per cubic meter. And so it then becomes a direct estimate of what is the shear elasticity, and that's the thing that is changing based on disease, injury, response to therapy. And that the shear modulus is related to the Young's modulus, and if it were that simple, that would be great. But the reality is, it isn't that simple. This is true if you have a homogeneous isotropic material with no damping in it, and you have no boundaries and reflections off the boundaries. Then it's very simple. I just need to get the wavelength, and I can estimate the shear modulus and actually how the waves are traveling. I can assume is governed by this very simple wave equation here. But that's not the case, because tissue actually is not elastic. It's viscoelastic. Depending on the application, it may or may not be isotropic. It may or may not be homogeneous, and depending on the size, we may have to worry about boundaries. So one thing is it's not elastic, it's viscoelastic. So there's all types of models. These are like using the idea of a spring and a damper to account for the viscosity. All types of models are used for this in tissue, and we found that what works best is something that's called a spring pod. Has anyone heard of a spring pod? Has anyone heard of fractional calculus? All right. Yeah, fractional calculus, I never heard of it until my colleague at UIC, Richard Megan, he has like a book on it, Fractional Calculus in Bioengineering. It's the it's an observation that mathematically, fractional calculus has been around for hundreds and hundreds of years. And it's basically the idea that you can take derivatives that are not integers, like not, like you don't think of a first derivative, a second derivative, you take a derivative that's raised to a fractional power. It sounds kind of crazy, but the math is actually pretty straightforward. In the frequency domain, it's simple. It's as simple as doing integer order derivatives. And what has been found is that these types of models can capture how the viscoelasticity in muscle in tissue is better than some of these other models using fewer parameters. So that's in one of my alternate titles. I use the word fractal and fractional. It comes from this because fractional calculus also can capture the fractal structure of viscoelasticity in tissue, in that you have fractal means you have different length scales, similar relationships across many different length scales, and that's called like a fractal structure. Fractional calculus will capture that in a simpler mathematical framework. So that's one aspect. Another thing is, if we have viscoelasticity, the waves are traveling at different frequencies, and at different frequencies the wave speeds are different. And so what this means is that you can talk about a phase speed, how fast a wave is traveling at a certain frequency, or you can talk about a group speed. If you have like a wave packet that creates a certain shape, it may be traveling at a different speed than because the components of it are all traveling at different speeds. All right, so this is getting a little bit in the weeds, but this becomes a challenge because some methods like MR elastography are directly giving you phase speed. Ultrasound elastography methods are typically giving you a group speed. You need to know how to get between the two. Okay, some other particular challenges with muscle. Muscle undergoes activation. So you tense your muscle, and this is kind of complicated here, but basically what this is showing is that we had a very simple model that captured the behavior of muscle that passively pretty well, but as soon as it was activated, we realized we need to go to a different type of model. All right. Another challenge with muscle is that it is certainly not isotropic or homogeneous. It's based on a fiber structure. It's actually fibers within fibers, kind of a fractal structure. And you're going to have different properties along the fiber versus across the fiber. All right, so this is something we can model or approximate as something that's called transverse isotropy. So no longer do we just need one shear modulus or one Young's modulus for the tissue. We actually need shear and Young's moduli both parallel to and perpendicular to the fiber structure. And so it ends up, really, we need basically three of these parameters to accurately describe the shear elastic properties of the tissue. So now it depends on which direction you're measuring the waves will affect what results you get. So another factor is that this is the challenge with muscle and with the cornea actually, is that it's under tension. And so even particularly skeletal muscle or cardiac muscle is fibrous. It's almost always under tension. And that tension, this is an expression for the wave speed. That the wave speed will depend on the shear elasticity, and it will also depend on the tension or the stress that the tissue is under. So then the question is, how do you differentiate what is causing what? And also you have different types of shear waves that will propagate depending on what we call polarization. You have to be worried about not only about the direction of the wave traveling relative to the fiber, but also the direction of the oscillations, the transverse oscillations relative to the fiber, both of those. So all these things make it a little bit more complicated, but also arguably could lead to being able to get more information, because that stress, which affects wave propagation, is also important and also altered by disease, injury, response to therapy. But we need to tease those apart. And now this is just a simulation showing the importance of boundaries. If I have a vibration source here that's sending waves out and it's far away from a boundary, you got a nice pattern here. Maybe I can get these wavelengths. I can figure out what are the properties this way versus this way. But if I have a boundary, you get a completely different type of wave pattern. And so these are called waveguide effects. And so you need to take those into consideration. And then here I'm showing, you also throw in the pre-stress. It gets more complicated. And these are kind of the same thing, but looking at different polarization of the waves, but still also having the boundary effect problem. Okay, so I came at this, so a lot of my training was more traditional vibrations, acoustics, I came later to medical applications. And I think in some ways I had a little bit of a different skill set or background than many people who were trying to do elastography, who were trying to use that first simple equation I showed, which just related wavelength to shear elasticity. I looked at this and some of these applications. I was working with, collaborating with folks at Northwestern University and at Rehab Institute of Chicago, now called Shirley Ryan Ability Lab. And they were showing me all this data they were getting on muscle in terms of waves traveling on the muscle and how it was being altered by the tension or the load put on the muscle. And I was looking at it, and you look in the literature, and you see so many publications that will say, my gosh, as we put tension on the muscle, the elastic properties of the muscle drastically change. And then I looked at it, and I was like, I don't know about that, because I was thinking of like a beam or a string. Think of your guitar string. You put tension on the guitar string to change the waves on it. You change the frequency because you're changing what standing wave pattern will be set up on that string. The more taut you make it, the longer the waves, the higher the frequency. So, and that is governed by a completely different equation. And that equation, so I felt that the people were missing the point with this. And I'm maybe going to gloss over some of the math here, but it gets a little more complicated in terms of you take into account the boundaries, the dimensions. All these things will alter how those waves travel. And so, and then the question is, are we measuring material properties or the tension on the string? Thinking of muscle as a string or as a beam, well, it kind of depends on the application. I think with muscle, we can treat it more like this, whereas the original application, or the application of elastography that has FDA approval is now, insurance pays for it because it's the preferred way to do it, is to stage liver fibrosis. The liver is this big chunk of tissue here. Maybe you don't have to worry so much about these tension effects, and you can just send vibration waves into the liver, and you can get a pretty good estimate of how stiff the liver is. Not the case with muscle. All right, so my colleagues actually, they did an experiment where this is a cat soleus, a leg muscle in a cat, where they could have measure how much tension they're putting on it. They could also activate it, and then they were using ultrasound elastography to track the wave propagation along the muscle. And what this is basically showing is that if I model the muscle as a string and then look at how much tension I'm putting on it, my model of it as a string almost pretty much exactly captures what we measure. So what this shows is that there's really no change in the elastic properties of the muscle. It's just the tension that you're measuring. Now, if the muscle gets activated, though, it gets a little bit more complicated. And this is part of an ongoing NIH project that we're working on, is to try to come up with better models for this to understand what is going on. Okay, so part of this work has been, if we want to adapt this to from simple models into something that's going to be useful in vivo, is we need to understand the muscle structure in vivo and be able to use that in our inverse part of the elastography challenge. So this is one of my former students. He was a really good MD PhD student. He was working on ways to take MRI images, not just anatomically of the muscle, but also using something called diffusion tensor imaging in MRI, which tells you the preferred fiber structure, the fiber orientation. And so we could infuse the fiber orientation into a anatomically accurate finite element model of the muscle. And then we could use that finite element model in our reconstruction, in our inverse problem. And so this is kind of just some shots of how he was trying to do this in terms of getting the images and then using diffusion to give you this kind of mapping of the fiber structure, and then use that to create a locally varying direction but transverse isotropic model of the muscle tissue, and then use that in elastography. So we did some experiments on exercise muscle, and we had to coming up with different ways to induce vibrations in the exercise muscle. This is done in an MRI. Working in an MRI can be challenging. This is in a high field MRI where we have a pretty narrow bore in which we have to make all this stuff work, and we have to do it using things that we can put in the MRI, so non-metallic structures. And in this case, we were trying to create concentric waves that travel towards the center. And then, well, this is actually our finite element simulation. This was the actual experimental data, kind of showing, yeah, we're getting pretty close in terms of how we were modeling this. And so then he did this on other things, even like chicken breast. There's a fiber structure to it that you can try to infuse into your model. And we were trying to see if we could simulate what we were seeing with elastography, and that's one of the reconstruction strategies is basically to iterate a finite element model until it matches what you see experimentally, and then you have the material properties based on whatever you used in your finite element model. Okay, so now let me change to a different application, which is the cornea. And for this, we use something called optical coherence tomography. And this is a fingerprint, somebody's finger. This is a high resolution optical method, which is going to give you a little bit of depth of penetration, but not much. And so now for all these imaging modalities, I collaborate with people. I'm not an OCT expert. I'm a wave mechanics expert. I collaborate with somebody for this. I collaborated with my colleague Dieter Klatt for the MR elastography. For the ultrasound elastography, I'm collaborating with folks at Northwestern University. All right, so this is an OCT image of this is of the retina, kind of showing that it has high resolution, and you can see the different layers of tissue in the retina. Our focus is on the cornea, though. This is an OCT image of the cornea, which is translucent, but there's also enough reflectors within the cornea that you can pick up the structure of the cornea. And so there are a number of groups that are trying to apply elastography to the cornea. And again, going back to one of my earlier slides, they want a non-invasive, non-harmful way to assess and map the cornea stiffness. And so this is one strategy is they'll try to apply some vibrations to it. The early methods, it was actually taking a little thing and kind of poking your cornea on the side, vibrating it. Not painful, but a little bit, a little freaky. Some of the newer methods, they're just using a very focused pulse of air to excite vibrations on the cornea. And so this is the type of image then we get from applying these vibrations, and then we'll see waves traveling on the cornea. Now the cornea is, this type of research has been going on for about, 15 years or so. the groups that have been leading this, they recognize early on that because of the thinness of the cornea, you can't use that simple equation that assumes waves traveling in a bulk media. And so they've always taken that into consideration, and these are what we call guided waves on a thin plate or on a thin shell. And so it's a little bit different set of equations, but still you can trace it back to the shear elastic properties. More recently, some groups observed that you do need to account for the fact that the cornea itself is transverse isotropic. It's made of layers. The stiffness going through the layers is very different than the stiffness in the layers. So you need to take that into consideration when you try to do like an inverse modeling of these wave fields to identify the mechanical properties of the cornea. So let's see, these are just more images here of the method. And for this, the muscle stuff I was showing earlier was down in the hundreds of hertz. The cornea is a smaller target. We need shorter wavelengths, so we typically will go to some higher frequencies to get those shorter wavelengths. Now, what has been observed in the literature, here again, I've been doing the muscle work had been doing it for a number of years, and then I was approached by a colleague in ophthalmology who was looking at wanting to do elastography on the cornea, and I was looking at the literature, and what I found here in the literature is that, oh my gosh, there's all these articles reporting how dramatically the mechanical properties of the cornea change depending on the intraocular pressure. So intraocular pressure also is something that can vary as a function of different diseases. So how do you then tease out a change in IOP versus a change in the properties of the cornea? Well, they said, well, the IOP is causing the changes in the properties of the cornea, but I looked at it and it reminded me of what I had seen with the muscle. We know it's under stress, and so let's take a look at that. And so yeah, we can do some basic modeling of intraocular pressure creates a normal stress in the cornea. This is a simplified model here of the whole cornea and the whole sclera, the whole globe of the eye. And we can kind of study that, and if we incorporate that into these complicated equations, which are also accounting for the transverse isotropy of the cornea, we find that we get a different overall estimate. If we measure waves traveling on the cornea, we're going to come up with different values. So yeah, here again, the basic idea, you may have the tissue stiffens and the cornea tissue stiffens, yeah, the wavelength's going to change, but also because of the pre-stress, it's going to change even more. All right, and so these are images taking up, we're measuring, this is a way of looking how things change as a function of the frequency, and these are experimental measurements, and we were doing simulations trying to understand this by fitting our model that also accounts for stress. The intraocular pressure created stress in the cornea into the modeling. And basically what this shows is that what was published in the literature for these studies on the cornea was that at different intraocular pressures, which they typically report those in millimeters of mercury, they were measuring different shear moduli for the cornea. If we take into consideration the fact that the intraocular pressure is creating a different stress field in the cornea, it modulates what those values of the shear elasticity are significantly. Now we're still reporting changes because tissue is nonlinear in the sense that as you stress it, you will potentially have changes in its properties. But that's separate from the changes that are in how waves propagate that are caused by the stress in the tissue. Okay, so this was published, I guess, about a year or two ago, showing this effect. Okay, so that is, I'm being pretty quick, I guess, on my technical presentation. And then I can talk a little bit about my department. But any questions? Yeah, sure. Go ahead. Go ahead.
I have a question on your the corneal applications.
Yeah.
Was there any factor that was used to correct for the frequency that the probe was using to probe the cornea versus the spatial or temporal covariance of the machine, the OCT machine that was imaging the cornea? How? Because I guess you're looking at waveform propagation in the corneal layer, right?
Yeah.
So what was, I guess, what was the correlation between the frequency at which the probe was probing the cornea to the spatial covariance or the covariance of the machine?
Well, we certainly, for the cornea, actually we did two. If you're doing like the puff test, that's like an impulse. And what we're doing is we're applying a Fourier transform, and we're looking at things on a frequency by frequency basis. I guess in terms of, so we definitely do take into consideration what frequency, because we know things will change with frequency. In terms of how that might interplay with how the OCT is operating, I have to ask my OCT collaborator, but I think they're operating at dramatically different frequencies, and so it's not a factor. It's like, it's similar with, I want to say it's kind of similar with like ultrasound elastography. Ultrasound is, you're sending in compression waves at anywhere between 1 and 50 megahertz. That's 1 in 50 million vibrations per second. And those compression waves travel in, they bounce off the different layers of tissue, and you get a reflection back. Now, if that tissue is moving, you have the Doppler effect, where it's going to shift that frequency a little bit, and we're just picking up that slight shift in the frequency in order to assess how it's moving. But I think for the optical, I don't think we're really using the Doppler effect. It's more a tomography type thing, or just distance there.
Right. So you're not just taking one image, right?
Right.
It's multiple different images you're taking and using that.
Correct.
Okay.
Yeah, for sure. Yeah, with all these techniques, you are definitely taking multiple images. And then even for the MRI, you are you're offsetting. You're taking snapshot images, and you're offsetting the phase of those images relative to the phase of the vibration that you're inputting. And then based on those, you can kind of reconstruct how a tissue was moving at a particular location. And then you got to map that out over a whole region. And so actually there's MRI is very complex in terms of how it's done, but there's many different what they call pulse sequences to do it. But for elastography, a lot of times the challenge is trying to do this as fast as you can. If you did it to get all the information you wanted, you'd have the person in the MRI for an hour. That's not good. So you're trying to get it down to minutes. And at best, you can get it down to maybe 5 to 10 minutes or so, that you really want it to stay still, right, while you're sending in these vibrations, and you're doing all these phase offsets, and a lot of other things to make that happen. So, yeah. Other questions? Yeah.
Thank you for that technical talk. I really enjoyed it. So when you are backing out the true mechanical properties of the tissues, and you run into this active contractile state in the muscle where you cannot put some sort of load sensor to know what that load is, versus in the cornea where you know the intraocular pressure. I assume there is no active contractile cells in the cornea. Why is the knowing the intraocular pressure and some sort of geometry of the cornea not enough to back out what the stiffness is, versus in the muscle you don't know what those forces are, but it seems like in the cornea you have the intraocular pressure?
That's a good question, and actually I would say if you want to be truly non-invasive, you don't have the intraocular pressure. It's an unknown. Mr. Ellis reminds me, if you ever go to the optometrist, they do those puff tests. They're puffing. They're trying to get at your intraocular pressure, which to me was kind of ironic, in that people doing elastography were assuming that the intraocular pressure, I don't know, they were trying to ignore the effect of the intraocular pressure, whereas that puff test, that's all about ignoring that your cornea might be different and assuming that any changes in how the cornea deforms are entirely due to the intraocular pressure, where it's not. It could also be due to changes in your cornea. So I'm a little skeptical of how much information you're really getting out of those puff tests. But your question is an important one, and that is the challenge right now, is if you want to do this truly non-invasively, either for the cornea application or for the muscle application, you can't. You have no idea of knowing what the actual loads are. And so our strategy right now, I'm funded to work on muscle. My cornea stuff is kind of dormant for the moment. But what we do is we're saying, well, we need to put the muscle under different load conditions, try to have it completely unloaded, and then do different passive loading conditions, and then also do muscle activation conditions where you try to train the patient to figure out what is 30% total muscle activation, 50%, based on some kind of electromyographic feedback. And then we're hoping that if you have this set of measurements, you can then basically do some type of interpolation and be able to separate out what is caused by stress versus what is caused by actual changes in the material properties. Because the material properties are actually also changing when you put something under stress. So, but that is the challenge. I guess that's the key, aim of our grant. And that's why NIH gave us money, was the recognition that nobody's really solved this problem.
Maybe a little counterproductive. Could you then, once you solve all the passive properties of the tissue, could you then back out with contractility? If you knew what the basal, passive state of the tissue is?
Yeah, basically that is our approach, is we need to solve the passive problem first. And then, once we have that down, then we can start trying to tackle the muscle activation problem. Yeah. And for the cornea, I have this one. I worked with another, it's a really good MD PhD student. He was looking at, this is frequency versus wave number. These are dispersion plots. And so from these, we're seeing how the wave behavior changes as a function of frequency. And we have the red line is a model. The color scale in the background is experimental data. And it's showing there is some blurriness to the experimental data. There's signal to noise ratio issues. But what we had observed is that maybe we could look at different frequency ranges, and at certain frequency ranges, like on the lower end, we saw much greater sensitivity to the IOP, the intraocular pressure, versus at the higher frequencies, the intraocular pressure had less effect. And so the thought there was that if we use the full frequency range, we ought to be able to tease apart what's caused by the IOP versus what's caused by changes in the actual corneal tissue properties. Yes?
So you have a heterogeneous tissue.
Yeah.
What's kind of resolution how small can some of the inclusion in that tissue be for you to be able to detect it? And I assume that's a function of the differences in moduli between the two materials as well, right? So
it is, yeah, but it is, and it's also a function of modality. For the OCT, yeah, we were down to micron, really tiny resolution. But there's a question of pixel resolution, and then there's a question of how the resolution of identifying material properties. So obviously OCT, incredible pixel resolution. Ultrasound is if you're near the surface, you can get pretty high resolution. But at deep penetration, your resolution gets worse. And then with MRI, it's the same resolution throughout the body, more or less, but it's not it's now we're talking at best on the order of millimeters for pixel resolution, maybe even centimeters. But then you impose the waves, the vibratory waves, and so you want to go to high frequency, short wavelengths to give you better ability to discern changes in material properties. But as you go to higher frequencies, the waves attenuate so rapidly that it becomes a real challenge. So a lot of the focus then is like creating, using, we call it modulated radiation force of ultrasound, even in MRI, to send compression waves into a deep point, but then have them modulate a little bit to create a sharp little vibration inside the tissue and see waves around it and try to image it. But bottom line, to get to your question, I think at best we're millimeters to centimeters is what we're looking at. And so we're not going to be able to detect small tumors, small scattered tumors. It could be larger tumors or over like in the liver. You're just trying to get an overall assessment of the properties. And there is a lot of work in looking at blood vessels and elastography of blood vessels, the aorta, some of the other major arteries, as well as the heart. And I did some work on heart elastography a while ago. We did it actually on a mouse heart, which beats at 500 times per minute. And the mouse is breathing about 100 times per minute. And there we had the challenge that we tried to gate. We're sending in vibrations at around 600, 700, 800 hertz. And we're trying to gate those off. We're certainly gating them with the respiratory cycle, and we're tracking the cardiac cycle so that we can recognize the fact that things will dramatically change because the stress environment changes in the heart. So that was very technically challenging. We published it, but I haven't done anything with that since. Yeah. Other questions?
I have a question. So in translating this to a clinical environment where it could be used for some type of diagnosis, so kind of two parts to this question. One is sort of how close and what needs to be done in order to get there, from a scientific standpoint, and then also from an engineering standpoint of the devices and the instruments that need to be designed to implement, the waves. And so, do you have any thoughts?
Well,
on that?
yeah. I mean, in terms of clinically, elastography is done on the liver now, both using MRI as well as ultrasound.
Well, I was, so like muscle and cornea, that like what you're.
Yeah. I think the cornea is close in terms of translation. I mean, there are a few groups that are really pushing, trying to translate it. I don't see issues. A lot of the issues I think are just clinician-physician buy-in.
Sure.
In terms of the method, and then having to do the clinical trials to show its
So in
value.
terms of the instruments that are needed to actually interact with the eye and create this pressure, and are there any opportunities there for engineers to develop, better technology?
Yeah, I think, there certainly is in terms of, packaging something that was built from pulling stuff together in the lab into something that.
Because the initial clinical trials will be based on, it won't be based on a commercial device that a company is trying to
Not the initial.
develop, right? It'll be based on, laboratory, and then, because obviously you gotta demonstrate the applicability first before a company or anyone is gonna get one of our terrific students to say, here, design me a device that I can start, selling and get through FDA approval.
In terms of like the MR, elastography, it is FDA approved for the liver. The next thing it'll probably get FDA approved for, I think, might be the brain. I'm not sure. that's where a lot of the work is. A lot of the research is on the brain. Is your brain stiffness varies with your age, your gender, and also things like neurodegenerative diseases like Alzheimer's, even multiple sclerosis. They've shown that there's measurable differences. The question is, what's the value sometimes of, that's always a question. What is the value of being able to measure something? You have to be able to detect it early so that it might alter treatment differences if there is a treatment.
Right, just knowing an answer, if you can't affect the treatment, doesn't matter. Yeah, as a matter of fact, I was at just kind of coincidentally at BMS, someone invited me to MRI elastography session, which it's not my area at all, but he was giving a talk and he invited me to come here and talk. And that was on brain. Most of it was focused on brain, and so there's a device that rattles the brain. And his particular talk was on trying to use inherent movements because of the blood pressure on the brain, which is obviously a very different frequency, but then doesn't require rattling the brain.
Yeah, right. And there also you can look at porosity issues and looking at the inflow and outflow of cerebrospinal fluid in the cranial chamber. And so maybe you can use the same techniques to pick up important information from that as well as some aspect of elastography. So there's a company, Resounded, that came out of the Mayo Clinic. The inventor of MR elastography is Richard Eman and Armando Manduca. In 1995, they published a paper showing how they can do MR elastography. So it's not that old. Well, I don't know, 1995. I don't think that's
It's all relative.
That's all relative. But they have a company that is FDA approved for the liver, and they sell a device that basically it's a pillow. That you use a big loudspeaker that sends pulsating air waves through a conduit into a pillow that you can wrap around your head, put up against a drum like that you put up against your side, and it works pretty well. They've shown consistency for these larger targets. The more challenging ones are when you start to get into things like the muscle, where you have stresses, significant stresses, or vascular, where you have waveguide effects that you need to take into consideration. But it's been applied to pretty much every organ in the body now. Yes?
Hey, I really enjoyed your presentation, by the way. I have a question. So how do you see the technique, like elastography, contributing to like our further understanding of like how these neurodegenerative diseases like start, like the onset, and like neurovascular coupling? Like, can we incorporate that into like further study?
Yeah. My one colleague, Dieter Klatt, does a lot with Alzheimer's, and I think there's a lot of debate in the Alzheimer's research community in terms of what's causing what. You're measuring changes. What comes first? I think is what some of the debate is. I'm not an Alzheimer's expert by any stretch, and so part of the argument to use elastography is that it's another tool. That they do a lot of there's mouse models of Alzheimer's, to try to do a lot of studies to longitudinally look at what changes you might see first, not just using elastography, but other types of MR-based imaging methods. So I don't have a concrete answer for you other than that. That's something that's ongoing research. Yes?
Is there a relationship between like the fiber damage you see like after maybe exercise or a therapy session and the like readings elastography gives you?
Yes, there is, for sure. And so when studies are done on muscle, you have to be consistent in terms of having an individual, like no serious weightlifting workouts within several days prior, because you are tearing your muscle, and then hoping that it'll grow back stronger. And so definitely there is an effect. Yes.
So like it has a lot of applications in like you can test out how effective a therapy was, like a therapy session.
Yeah, you could potentially. Yeah, I think that is certainly a possibility. Yeah. Or how effective or ineffective a workout
to anyone to talk about your career development just a little bit. Just in terms of, like you said, you were in vibrational mechanics before. Can you talk a bit about that transition from that to sort of a different sort of field?
Yeah. No, I went to Ohio State University. I'm actually from Cincinnati, Ohio. He just said to step out. But we didn't have biomedical engineering back then, and actually I got interested in acoustics and vibrations, and eventually I spent some time. It's interesting. I had a fellowship as a grad student, O&R fellowship, and I spent some time at a Navy lab, or Navy research lab in DC, doing acoustics, and I got really interested in active sound and vibration control. I came to UIC in 1995 in mechanical engineering, and then a couple things happened. I started talking with my colleagues in biomedical engineering. One in particular, actually it wasn't the colleague, it was the student. It's the students that he had a grad student who had heard about MR elastography. It had just come out, and he wanted to try it. And his advisor in BME, Richard Megan, was an expert in MRI, but knew nothing about elastography, and so he was looking for somebody who knew something about acoustics and vibrations. So the student contacted me. I started collaborating with Richard Megan and started to make that progression. At the same time, I remember I went to, because there's these opportunities to go work in Navy labs in the summer as a faculty member. I went back to the Navy Research Lab in DC and was working with the acoustics group there on basically submarine acoustic stealth, using the idea of putting a smart skin on a sub that would sense incoming sonar and be anechoic, no reflections back. While I was working there, though, there was a guy there who was just a brilliant acoustic theoretician, and he was starting to work on MR elastography in terms of the reconstruction problem. He had been contacted by the group at the Mayo Clinic. It was kind of weird that I was seeing that and just seeing opportunities at my home institution and seeing all these opportunities. And so I started migrating into that. Also, another thing I didn't talk about is I've done a lot of work in pulmonary acoustics. And I was contacted by physicians in Chicago who were trying to develop new acoustic diagnostic methods, and they wanted somebody who knew acoustics, so I started collaborating with them as well. And that's like a whole other line of research I had. So it was through those collaborations, I started getting more involved in that, and then moved over to biomedical engineering after having been at UIC for 14 years, and kind of haven't looked back. I've entirely focused on medical applications since then. So yeah, that's kind of my progression. Yeah. It's a different landscape these days. So I don't know how we
still got another couple minutes if anyone wants to. I'm not done before everyone to class, but any questions?
I can, I was gonna say, I have, really quickly, a few slides on my department.
Sure.
Already seen that. Where am I?
I just want to show we have some different areas. So we have this bioinformatics, quantitative biology area where we have a lot of very successful researchers. This is AI, your AI and everything. These guys have been doing AI for 20 years or longer. And so this is a separate degree program. We have researchers involved in cell and tissue engineering. Pointing this out in terms of anyone looking to relocate to Chicago, lots of opportunities. These guys are looking for grad students. And then, of course, imaging, which I've been talking about. A lot of optical imaging. This Zin Chen was my collaborator on the OCT, optical coherence tomography, and Dieter Klatt was my collaborator on the MR, elastography. And then we have a lot of people who do work in microfluidics, diagnostics, we call liquid biopsies, point of care sensors. Ian Popowski, very cool technology, take a drop of blood, find the 1 in a billion tumor cell circulating in the blood based on its different inertial properties. And then we have this collaboration with the Rehab Institute of Chicago, now called the Shirley Ryan-Ability Lab. It's the #1 rehab hospital in the country. My faculty, their labs are there, but there are faculty. This is how it's a nice facility, basically, where their students will do research at that facility, although they're UIC students. And so that's mainly, Jim Patton and Ming Wu. And then I have a few other faculty here who do research, different types of research in that area. And that's pretty much it. Just point out the different, a lot of my colleagues are looking for grad students, if anyone in the future is tired of the nice warm weather. It's not that bad, but I'll end there. So, thank you.
See you all in the closed session.